कैसे सिंथेसिस होता है पहले वो देख लेते हैं, ताकि आपको थोड़ा सा इजी हो जाएगा उसके बारे में पता करने के लिए। ओके, तो हमारे पास डीएनए होता है और डीएनए की जो लेंथ होती है अप्रोक्स, तुम लोगों ने 12th में पढ़ चुके हो कि भैया डीएनए की लेंथ कितनी होती है, अप्रोक्स 2.2 मीटर। ओके, एंड जो सेल है, ऑब्वियस सी बात है कि जो डीएनए है इट प्रेजेंट इनसाइड द न्यूक्लियस, डीएनए प्रेजेंट इनसाइड द न्यूक्लियस। ओके, बट जो न्यूक्लियस है, न्यूक्लियस की साइज इन 10 to the power minus 6 meter, that is in millimeter. Okay, so there is large size difference between the DNA and the nucleus. So how it is possible for the DNA to be in the nucleus, to be in the nucleus? So nucleus के अंदर कैसे DNA रहेगा, उसके लिए हम देखते हैं आगे की क्या प्रोसेस करता है। तो अब ऐसी बात है कि हमारे पास कोई एक धागा है, we have a okay, we have this much length of the wire and we have to put it into this, we want to put into this. So we have one method to fold this wire, we can fold this wire in this fashion and we can put into this. So same thing DNA will use, and how, but how the DNA will fold itself, DNA खुद को कैसे फोल्ड करेगा और बेसिकली फोल्डिंग और पैकेजिंग कैसे होगी डीएनए की। तो as you all know कि भाई डीएनए के पास होता है नेगेटिव चार्ज, डीएनए हैव द नेगेटिव चार्ज। Why negative charge? Due to the presence of phosphate group H3PO4 नेगेटिव। फॉस्फेट ग्रुप की प्रेजेंस के कारण डीएनए के पास होता है नेगेटिव चार्ज। ओके, एंड हमारे पास एक और प्रोटीन होता है जिनको हम बोलते हैं हिस्टोन प्रोटीन। एक हमारे पास प्रोटीन है जिसको हम बोलते हैं हिस्टोन protein, and this histone protein, it is made up of the histone protein made up of the arginine and lysine residue, arginine and lysine residue, and both are the basic amino acid, both are the basic amino acid, and basic contain the positive charge. That's why the histone protein contains the positive charge. Histone protein has two structure, one is the histone octamer, this is the histone octamer, and this is the H1 histone. Okay, so basically I told you about the histone and all this packaging of the DNA, but for the easy understanding and explaining it again for you. Okay, so this is the H1 histone and histone octamer, and it contains the positive charge. So DNA has the negative charge, histone protein has the positive charge. There will be the wrapping of the DNA, this is the DNA over the histone protein. Simple, histone protein. Okay, why histone protein is positive charge, DNA has a negative charge, there will be wrapping of the DNA on the histone protein, and this structure is formed in known as the nucleosome. And again folding will be there like this. Okay, again folding will be like this, and this will be the chromatin material, and at the end we will get the chromosome. At the end we will get the chromosome like this. So just assume that okay, so like this we will get a chromosome, but after we will get a chromosome, there is the primary constriction. Primary constriction. What is the first thing here? We will get a primary constriction. So primary constriction. Now, primary constriction results into the formation of a structure known as the centromere. Centro Centromere. Okay. So here will be a primary constriction like this. So here will be a primary constriction like this, and this structure here is known as the centromere. This is the centromere. Okay, this structure known as the centromere. It is the site of primary constriction. Okay, and centromere has a disc shaped around it. Centromere has something disc-like structure around it. This disc-like structure around the centromere is known as the kinetochore, and this is the proteinous covering of centromere. Proteinous covering of centromere, disc shaped around the centromere, the kinetochore. Please remember this thing because it is also important in your next chapter. But for now, it is the centromere which is formed due to the primary constriction. Kinetochore is the covering or proteinous disc-shaped covering around the centromere. So it is known as the kinetochore. Okay. Now, on the basis of the centromere, chromosomes are classified into four types. On the basis of on the basis of centromere, on the basis of centromere, chromosomes are classified. Chromosomes are classified into four types. First is the telocentric, acrocentric, metacentric, and submetacentric. So, in the telocentric, acrocentric, metacentric, and submetacentric, first we'll talk about the metacentric. In the metacentric, the centromere will present it, will locate it, the center of chromosome. Okay, so the centromere will present or locate at the locate at the center of the chromosome. Like we have a chromosome. In this situation, you can see the centromere is located at the center of the chromosome. So this type of the chromosome is known as the metacentric chromosome. If the location of the chromosome, the location of the centromere shifted to the near to the center, assume it will shift to here, then it is known as the submetacentric. So centromere will present near to the center, near to the center of chromosome. It's not the submetacentric. Then acrocentric or telocentric. So telo means terminal, T for telo and T for terminal. So when the centromere is terminal like this, so the centromere will present here. It is known as the telocentric. And if the centromere will be towards the terminal, it is known as the just near to the terminal point. It is known as the acrocentric. So this is the acrocentric. telocentric, submetacentric, metacentric. Okay, I am drawing the location of, this is the location of centromere on the chromosome. So if the centromere present in the terminal site, it is known as the, it is terminal centromere, acrocentric, subterminal centromere. Okay, so this is the telo acro, sorry, telo acro, submetacentric and metacentric. It is about the different type of the chromosome on the basis of the location of centromere. Next topic is the okay, one more thing is there. We talk about the primary constriction. Okay, cons primary constriction. What about the secondary constriction? Okay, just assume here this is the centromere. Okay, and here we have the primary constriction. If there is again a secondary constriction, okay, here also present a secondary constriction like this, and here chromosome, and this side is known as the satellite. So primary secondary constriction. So what is the second satellite? Satellite is just nothing, just a secondary condition. Sorry, secondary constriction. It is nothing but a secondary constriction. Okay, so someone will ask you what is the satellite. So such second, in case of the chromosome, just in case of the chromosome, not in every case, but in case of the chromosome, satellite is just a secondary constriction on the chromosome. That's all. Okay, so it is about the centromere, type of the centromere on the basis of location. Okay, now we'll talk about the cytoskeleton. Cytoskeleton, cytoskeleton. What is cyto? Cyto means cell. Skeleton means structure. So who will maintain the cell structure? These structure are known as the cytoskeleton. So there should be some structure which will be therefore therefore maintaining the structure of cell. Okay, कुछ स्ट्रक्चर होंगे जो कि आपकी सेल के स्ट्रक्चर को मेंटेन करने के लिए बने होंगे, उनको हम बोलते हैं साइटोस्केलेटन. उसमें आते हैं आपके माइक्रोट्यूब्यूल्स, उसमें आते हैं आपके सिलिया एंड फ्लेजला, सिलिया एंड फ्लेजला, एंड वी हैव अनदर थिंग सेंट्रोसोम एंड सेंट्रियोल्स. So microtubules are structures which are formed by these structures are formed by the tubulin protein. Tubulin protein. Okay. So these are just tubules which are formed by the tubulin protein. Please remember the word tubulin protein is very important. Okay. Now cilia and flagella. We'll talk about the cilia and flagella in detail. Important for NEET exam. Cilia and flagella. Structure is most important thing. We call this structure of this flagella is the 9+2 structure. Cilia flagella has the 9+2 structure. What is the 9+2? Okay, we will talk about it. So first of all, what is the cilia and flagella? Cilia and flagella, they are the outgrowth of the cell. They are the cell outgrowth. Cilia is smaller than the flagella size wise, and cilia has the function of it will move cause the movement of either the cell either the cell or the surrounding fluid surrounding fluid. And flagella, they are comparatively longer and responsible for the cell movement. They cause the cell movement. Basically, for the mobility or motility of the cell, the flagella is important. It is responsible for the cell mobility. I told you about it. And we have a eukaryotic cell like this. Okay, we have the eukaryotic cell like this, and we have the flagella. If the eukaryotic cell will have flagella, this eukaryotic or bacteria, basically, what is the eukaryotic cell? It's the bacteria. So, bacteria is motile. It can move with the help of the flagella. And we have structure like this. We have a structure like this. They are the cilia. They will use for the adherence to the host body. होस्ट बॉडी के बॉडी में अधेर होने के लिए यूज कर सकते हैं, या फिर ये यूज हो सकते हैं मूवमेंट ऑफ द फ्लूइड। यहाँ पर कोई फ्लूइड होगा, अगर यहाँ पर कोई फ्लूइड है यहाँ पर सेल के आसपास, तो फ्लूइड का मूवमेंट करा सकते हैं। ओके, तो ये है इनका फंक्शन। ठीक है, अगर हम इसका स्ट्रक्चर देखें तो दे हैव अ कोर। व्हाट इज द कोर? कोर इज द सेंटर पार्ट ऑफ द structure. So they have a core known as the axoneme, center part of the structure. Okay, just I'm drawing the structure of cilia and flagella. So they have a plasma membrane. They have a plasma membrane. Inside the plasma membrane, they have a core. Core is known as the axoneme. They have core that is called the axoneme. Okay, and okay, so the structure is 9 plus 2. The structure is 9 plus 2. Now I'm going to explain the 9 plus 2 structure. So we will have some tubules. Now again, microtubules are there. Okay, there will be microtubules. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So like this, we have 9 doublets. There are the 9 doublets of microtubule. Okay, yes, you are seeing the structure from the anterior side. Okay, this is the structure of the anterior view. Basically, the microtubule is running like this. Okay, on the back side you can see they are running like this. They are running like this. Okay, yeah, the microtubule running like this. Okay, but we are seeing the structure from the anterior view, from the front. Okay, we are seeing the structure of the flagella from the front. So we have 9 doublets of microtubule. They are the 9 doublets of microtubule. So doublets of microtubule. Okay, you can name them as B, A, B, A, B, A. Likewise, A, B, A, B. Okay, like this. So we have 9. 1, 2, 3, 4, 6, 7, 8, 9. They are running parallel to each other in a long axis. They are running parallel to each other in a long axis. axonobacteria and the okay, so they are the peripheral. Okay, they are peripheral, but we also have the central microtubules. Okay, central microtubules. Okay, so central micro, we have two central microtubules. Two central microtubules like this. One and two. So one and two central microtubules. Okay, and central microtubules are covered by the central sheath. Covered by a central sheath. This is a central sheath. Okay, so this is the central sheath, and they are connected by a bridge. They are connected by a bridge. So this is the bridge. Okay, so name we name this structure central sheath, which covers the central microtubules. So this is the central tubules. Central tubules. Central tubules are connected by the bridge. That's why the structure is named as the 9 plus 2. 9 peripheral and 2 plus 2 is the central. They are the central microtubules. And microtubules are made up of the tubulin protein, I told you. And they are the peripheral. Peripheral microtubules, central microtubules. Okay. And this this is peripheral microtubules are linked by the linkers. They are linked by the AC linkers. They are sorry, they are linked by the AB linkers. AB linkers. So they are linked by the AB linkers like this. AB, 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 AB linkers. Okay, so they are the linkers. Linkers connect the peripheral tubules together. And we also have radial tubules. We have for radial spokes. We have the radial spokes. So radial spokes like this. Okay. So radial spokes connect the they are the radial spokes. Radial spokes. They connect the peripheral and central tubules, or we can say microtubules. Okay, central tubules or microtubules. So radial spokes, and we also have interdoublet bridge. We also have beside the linkers, we have the interdoublet bridge. There, this is the interdoublet bridge. So this is the interdoublet bridge. Okay, so this is the structure of cilia and flagella. Cilia and flagella. I'm explaining it again. Core is known as the axonomy, and the structure basically function. Function is movement of the fluid around the cell or cell itself. So cell movement will be taken up by the flagella. And the structure is 9 plus 2 structure. 9 plus 2 structure, in which 9 is peripheral microtubules and plus 2 is the central microtubules. All the structure is covered by the plasma membrane. This is the plasma membrane. Okay, so this is the plasma membrane, and they are peripheral 9 doublets. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 over. 1, 2, 3, 4, 5, 6, 7, 8, 9, 9. Okay, 9 doublets and 2 centrally plates microtubules, which is covered by the central sheath. Central and peripheral microtubules are connected by the radial spokes. Doublets are connected by the linkers, AB linkers, and the in the inter doublet bridge. And central microtubules are connected by the central bridge. Okay, this is the structure of microtubules. They are arises from the flagella and cilia, and flagella arises from cilia and flagella arises from the basal bodies. Okay. Our next topic is the centrosome or centriole. Okay. Now we'll see about the centrosome and centrosome and centriole. For the centrosome and centriole, basically centrosome is nothing but a structure containing two centriole arranged at a perpendicular to each other. Okay, two structure arranged perpendicular to each other. Two structure arranged. There the two centriole. Okay, two centriole arranged perpendicular to each other and surrounded by a pericentriolar material. So here we have the pericentriolar material. Here we have the pericentriolar material, and they are the two centriole arranged theta perpendicular to each other, surrounded by a pericentriolar material. Pericentriolar material. Pericentriolar material, and they are the centrioles. Okay, 9 degree, one and second. So two centriole arranged at a perpendicular to each other and surrounded by a pericentriolar material is known as the centrosome. And the centriole has the cartwheel appearance. Centriole has the cartwheel appearance. Now talk about the centriole. We'll talk about the centriole. So what is centriole basically? Centrosome is nothing but a two centriole arranged at a perpendicular manner and surrounded by a pericentriolar material. That's the centrosome. But what is the centriole? The centriole has a cartwheel appearance. Cartwheel appearance. Cartwheel appearance. Okay. So the structure of centriole is known as the 9 plus 0 structure. Just similar to the this one, but there is no central microtubule. Okay. But here are the triplets. Okay. Okay. I'm drawing it. So 1, 2, 3. Okay, so nine is there is nine triplets. Okay, nine triplets. 9 plus 0 arrangement is present in the centrosome and centrioles, basically centriole, and we can name them A, B, C, A, B, C, A, B, C, A, B, C, A, C, A, B, and C, A, B, C, A, B, C. Okay, and there is the AC linkage between the triplet. AC linkage between the triplet. So what is the AC linkage? Just see here. There is the A and C. So A, C, sorry, AC again A, B, C, AC, 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 AC linkage, AC linkage, AC linkage, and AC linkage, and AC linkage. So this is the AC linkage in the microtubules. Okay, AC linkage in the microtubules. Okay, and this AC linkage, they are the triplets. Okay, they are the triplets. So they are the triplets of microtubules connected by AC linkage. Okay, AC linkage, and the central part is a proteinous part. Central part is a proteinous part, and the proteinous part. And this proteinous part is known as the hub. Proteinous part is connected by the peripheral tubules by radial spokes. Okay, so they are connected by the radial spokes. Okay, so this is the radial spokes. They are the radial spokes. Radial spokes. Okay, so we have the 9 plus 0 where there is nothing in the center. Okay, nothing in the center. We have the hub in the center, but that is not a microtubule. That is a proteinous part. So we will not include in the 9 plus something. 9 plus 0. 9 is the triplet, peripherally placed triplet, and there is nothing, no microtubules in the center. So here is the 0. So this type of the structure present in the centriole. And what is the centriole? And what is the centrosome? Sorry, what is the centrosome? Centrosome is the two centriole located perpendicular to each other and surrounded by a pericentriolar material. They also surrounded. The centriole also surrounded by the pericentriolar material. Okay, this is also surrounded by a pericentriolar material. Okay, so it is about the centriole, centrosome, microtubules. I think we completed the chapter. One more thing, microbodies. Microbodies are the membrane-bound vesicle structure which contain the different type of the enzyme basically and present in animal cell and plant cell. Microbodies, nothing but the vesicle-like vesicle-like membrane-bound structure present in plant and animal cell contain various types of enzyme. Okay, so important thing, microbodies are present in plant cell and animal cell both, and they contain various type of the enzyme. Okay, so we completed this cell, the fundamental unit of the life. Now we will start cell cycle and cell division. Okay, we will start a cell cycle and cell division. Okay, revise the today's lecture once again. We talked about the chromosome, types of chromosome, and then we talked about the types of chromosome: centromere, kinetochore, and telocentric, ectocentric, metacentric, subcentric. They are types of the chromosome on the basis of the location of centromere. Then cytoskeleton, cell structure maintaining substance, that is the cytoskeleton. They will, what is the cytoskeleton function? Function is maintain cell structure. Okay. In which microtubules, cilia, flagella, centrosome, centriole. Cilia flagella has 9+2 structure, and I told you about this structure. And the, sorry, this is the central structure and microbodies, and chapter is over. Let's start the next chapter, that is the cell cycle and cell division. Cell cycle and cell division. Now, what is the cell cycle and what is the cell division? As you know, we have a cell, we divide into the two daughter cells. Simple, this process is the cell division. Okay. A parent cell divides into two daughter cells, the cell division. But in this process of the division, there the various events are taken place. जब डिवाइड होती है कोई सेल, उसमें क्या होते हैं, डिफरेंट इवेंट्स होते हैं जो कि सेल के अंदर होते हैं. So cell cycle is just a different events which is present in the division of the cell. So sequence of events present in the cell when a cell, or you can a parent cell, when a parent cell will divide into two daughter cells. So what are the various events? What are the various events which are present in this cell? So there is nothing like this cell want to divide, so it will just divide. No, there are various events before the actual cell division. There will be various events before the actual cell division. So we will try to understand them. Simple. So cell division. Parent cell divides into two daughter cells. Okay. Now cell cycle. In the cell cycle, we have see in the diagram first. Okay, so this is the cell cycle in which we have up to this point. It is known as the interface. It is known as the interface. And from this point to this point, we have the M phase. Basically, what is the M phase? M phase is the, it may be mitotic or it may be meiosis. Okay, we are not talking about it now. We are not specifically mitosis, not specifically meiosis. It is the M phase where the actual cell division takes place. Actual cell division takes place. यहाँ पर हम कोई स्पेसिफिक बात नहीं कर रहे हैं माइटोसिस मेयोसिस की, अभी हम जब सिर्फ फेजेस की बात कर रहे हैं कि एक साइकिल में कौन कौन से फेज आते हैं. ठीक है. नेक्स्ट, the interphase are divided into three different phases: G1 phase, S phase, and G2 phase. This is the gap 1 phase, synthesis phase, and gap 2 phase. Sorry, gap 2 phase. This is so this will be the G2 phase. This is the gap 2 phase. Gap 2 phase. Now the M phase are divided into two phases. It is the one is the karyokinesis. Sorry, one is the karyokinesis. One is the karyokinesis and one is the cytokinesis. So first, this one is the karyokinesis. Karyokinesis. This one is the cytokinesis. Now karyokinesis is again divided into some phases like karyokinesis. Okay, so this is the basic structure of the cell cycle. You have G1 phase, S phase, G2 phase, and this from here to from this point to this point is known as the interface, and from this point to this point the M phase. M phase divided into two phase: karyokinesis, cytokinesis. Interface divided into three: G1, S, and G2. Okay, then just assume that here that a cell is dividing into two cells. Okay, a cell is divided into two cells, so it has to go through the complete cycle. Okay, it has to go through G1 phase, S phase, and G2 phase, then karyokinesis, then cytokinesis, then it will form two cells. Now, most important thing, the interface is the longest phase. Interface is the longest phase. This is cell divided into 100 minutes. A cell divided into 100 minutes. Then 95 minutes taken up by the interface. So why so? Because it is a preparatory phase. Interface is a preparatory phase. यहाँ पर क्या होगी? एक्चुअल सेल डिवीजन की प्रिपरेशन होने वाली है. ठीक है, जो भी सेल डिवीजन होगा उसकी प्रिपरेशन होने वाली है. क्या क्या प्रिपरेशन होगी? न्यूक्लियस डेवलप होगा, न्यूक्लियर मेम्ब्रेन डिसॉल्व होगी, एंड न्यूक्लियस जो न्यूक्लियर मटेरियल डेवलप होने वाला है, डीएनए डेवलप होने वाला है, उसका सिंथेसिस भी होगा. तो वो सब चीजें कहाँ पर होंगी? इंटरफेस में. और यहाँ पर क्या होगा? एम फेज में न्यूक्लियर डिवाइड होगा, क्रोमोसोम डिवाइड होंगे, साइटोप्लाज्म डिवाइड होगा. तो काइलोकाइनेसिस, काइनेसिस मींस डिवीजन एंड कार्यो मींस न्यूक्लियस. तो न्यूक्लियर डिवीजन मींस क्रोमोसोमल डिवीजन. In the cytokinesis, cyto means cytoplasm, Kinesis means division. Cytoplasm division will take place with the cytokinesis. Okay, so 95% time taken up by the interface of the complete cell division. Now we will talk about the each and every phases of the interface and then M phase. Basically, cell division, cell cycle is divided into four phases. See through a flow diagram, cell cycle divided into two phase, M phase. Sorry, first is the interface, and second is the M phase. Interface is divided into G1, G2, G1S, and G2. M phase is divided into M phase divided into two, karyokinesis and cytokinesis. Karyokinesis is again divided into different phases: prophase, metaphase, anaphase, and telophase. Cytokinesis is again divided. Cytokinesis of two types: cell plate method and cell sorrow method. Okay, we'll talk about them in the coming explanation. Okay, so we'll start with the G1 phase of cell cycle. Okay, just wait for 2 minutes. Okay, so first phase of the interface is the G1 phase. G1 phase okay. G1 phase corresponds to the okay. Just please remember about this diagram. M phase and this is the G1 phase. This is the S phase and G2 phase. Okay. So G1 phase it corresponds to the interval between mitosis and initiation of the DNA replication. So basically, S phase that is known as the synthesis phase. So corresponds to it is the phase between the S phase and mitotic phase. Okay, mitosis phase. In the G1 phase, the cell is metabolically metabolically active Cell is metabolically active, okay, but not dividing, but not dividing. Okay, cell is metabolic. All the metabolically process or all the metabolism in the cell are going on, but they are not dividing. But not dividing. Okay, so cell will grow in the size. Cell will grow in size. Next is the S phase. S phase is also known as the synthesis phase. The synthesis phase most important for the NEET exam. They will ask 50% question from the this phase, S phase. So in the S phase, there is the synthesis of the DNA material. Synthesis of DNA material. Now please remember one most important thing. There is the synthesis of the DNA material. Number of the chromosome will be the same, but number of chromosome will be same. For example, if you have 2C amount of the chromosome in the G1 phase, then it will become 4C amount of the chromosome in the S phase. But if you have diploid 2N, it will be 2N. Means if you have 46 chromosomes, then it will remain same 46 chromosomes, but the amount will be double. So the amount of DNA will be double, but chromosome number remains same. Okay, so this is most important thing. Please remember this thing. In the S phase, again I'm writing in bold letter. In the S phase, 2C become 4C, but 2N remains 2N. That is the most important thing. Okay. Now, one more thing is there. Centriole will divide. Centriole, centriole also duplicate in the cell. Centriole will also duplicate in the cell. Okay. Centriole will form two centriole. One centriole form two centriole. Okay. So the DNA replication takes place in the synthesis of DNA material takes place in the or DNA replication takes place in the nucleus. This thing is taking place in the nucleus, but the centriole replication taking place in the cytoplasm. Taking place in the cytoplasm. Okay. Okay. Next phase is the G2 phase. In the G2 or gap 2 phase, the synthesis of proteins like tubulin protein and cell growth is going on, increases. Cell growth increases. Okay. Now, all the phases of interfaces are completed. Now we will come to the okay. But most important thing is the phase known as the quiescent phase. A phase known as the quiescent phase. So it is a G note phase or quiescent phase. This phase is present in the cell. Okay, okay. See here. Okay, if a cell is going to divide, it will enter into the G1 phase. Okay, please see here. If a cell want to divide, it will enter into the G1 phase, and from G1 to into G2 phase, and from G2 sorry S phase, from S phase to G2 phase, and G2 phase to uh, M phase, and then it will be divided into two cells. Now this cell has two probability. One, it will mature into a new cell or differentiate into a new cell, or it will again enter into the G1. So it has two probability. Okay, I'm using sorry to make it more clear. So this cell has two. Okay, we have two cell. If this cell will enter into the G node phase, then it will again go into the S, then G2, and then M phase, then again it will divide. If the cell will enter into the G node phase, then it will not divide. The cell which is present in the G node phase will not divide. That's why it is known as the quiescent phase or resting phase. Resting phase. Cell in this phase will not divide. Will not divide unless until they on they are not called to do so. Unless until called on to डू सो जब तक उन्होंने ऐसा बोला ऐसा डिवाइड होने के लिए बोला नहीं जाए। अब वो क्या होता है वो भी हम आगे देखेंगे क्या कब बोला जाएगा उनको डिवाइड होने के लिए वो भी हमें देखना है अभी थोड़ी देर बाद में। सो इन दिस फेज रेस्टिंग फेज सेल विल बी मेटाबॉलिकली एक्टिव मेटाबॉलिकली एक्टिव but proliferatively inactive. Proliferatively inactive means they will not proliferate, they will not divide. Now, in the quiescent phase or G naught phase, cell will not divide until unless called on to do so. Now, let's take an example of the meristematic cell. A cell which divide. Okay, a meristematic cell become a permanent cell. Now, this permanent cell will have some functions. It will not divide. Now, this thing is known as the differentiation. This is known as the differentiation. If differentiation, if a meristematic cell will convert into a permanent cell, and this permanent cell will have the different functions. Okay, so this process of conversion of meristematic cell into a permanent cell is known as the differentiation. If this cell again it want to convert into the meristematic cell, it is known as the de-differentiation. Is known as the de-differentiation. After the differentiation, this meristematic cell can divide. Now it can divide by entering into the G1 phase. Okay, प्लीज रिमेंबर वन थिंग, अगर कोई सेल G1 फेज में इंटर कर जाती है देन उसको डिवाइड होना ही पड़ेगा. अगर कोई सेल G1 फेज में इंटर कर जाती है देन that cell have to divide, it has to divide, so it has to divide. Okay, so again if this cell wants to convert into the permanent cell, and this process is known as the redifferentiation. So there is three things: de-differentiation, differentiation, and redifferentiation. What is the differentiation? De-differentiation and re-differentiation. Okay, differentiation, differentiation, and redifferentiation. So when a meristematic cell become permanent cell, and the permanent cell become, so permanent cell is a mature cell which has the different function to play. This thing is the differentiation. If this permanent cell will lose the maturity, and again it will become the meristematic cell, and this is known as the de-differentiation. Okay, and in the third thing, if this de-differentiated meristematic cell, again if it will become the permanent cell, then it is known as the re-differentiation. Okay, so if a cell which is present inside the G0 phase which will not divide is the permanent cell. If it has to divide, it has to go through a redifferentiation into the meristematic cell. Then it can divide. Then it can divide. If it will enter into G1 phase, then it has to divide. So if a cell is present in the G0 phase, if a cell present into the G0 phase, if it wants to divide, it has to convert into the meristematic cell value de-differentiation process. Okay. Now we come to the M phase. Come to the M phase. Now, M phase of, now we are discussing about the M phase of mitosis. Mitosis is also known as the equational division. Equational division in which chromosome number remains same. So how it is possible chromosome number to be remains same in the daughter cell? For that we have to see the process. Okay, so mitosis has four phases. Four phases with the prophase, metaphase, anaphase, telophase. Prophase, meta, and telo. Now, first phase of the M phase, that is the prophase. In the prophase, when the cell enters, okay, prophase is the phase. Again, see this diagram. Here the G2 phase, S, G1, and here your pyrokinase prophase, metaphase, anaphase, and telophase. So this is the first phase, prophase. So we are talking about the prophase. So in this process, now we want to form the chromosome. Okay, now we want to form the chromosome. We have the DNA material. Now from the DNA material we want to form the chromosome. For the chromosome there will be condensation of the chromatin material. So first in this process, condensation in this phase, there will be condensation of chromatin material. Chromatin material, ओके, जो सेंट्रियोल एस फेज में डिवाइड हो चुके थे, जो सेंट्रियोल हमने एस फेज में डिवाइड किए थे, एस फेज कहाँ गया हमारा, ये एस फेज है। एस फेज में हमारे सेंट्रियोल डिवाइड हुए थे, सेंट्रियोल डुप्लीकेट किए थे एस फेज में। नाउ दिस सेंट्रियोल, they are the two centriole nucleus, they are begins to move towards the opposite pole of the cell. So centrioles begins to move towards the opposite pole. Okay, opposite pole. So basically we consider a circle, and this is the equator of the circle. A line passing through the center of the circle is the equator of the circle, and we have two pole, upper pole and lower pole. Centriole will move towards this and this pole. Okay. Next thing, so how will we will make the completion of the prophase. So at the end of the prophase, at the end of prophase, what we will observe in the cell, the chromatin material, chromatin material condensed to form chromosomes. Now chromosomes are containing sister chromatids like this. They are known as the chromatids. They are the chromatids. Okay, chromatids. They contain chromatids. Initiation of assembly of mitotic spindle. Initiation of assembly of mitotic spindle. Mitotic spindle. Spindle fibers are formed. Okay, so this is the spindle fibers. Basically, I will tell you in the diagram. Okay, so and we will not observe any type of the Golgi apparatus. We will not observe endoplasmic reticulum, nucleolus. Nothing will be visible at the end of the prophase and nuclear envelope. So all will be removed. Nuclear envelope is not visible. Okay, so cell will look like this. Just assume here we have a cell. Sorry, we have a cell, and in this cell we have we have the chromosome. Now chromosome, sorry, nucleus, okay, nucleus, and this nucleus, inside the nucleus we have the chromatin material. This is the chromatin material. This chromatin material become compact. This chromatin material become compact and form the chromosome like this. It forms the chromosome like this, and at the end the nuclear envelope will disappear. The nuclear envelope will disappear like this. Okay, and the centriole will be present at the two pole of the cell, upper pole and lower pole. So upper pole and lower pole, because there are two centrioles, and this centrioles begins to form spindle fibers like this. They are the spindle fibers. Spindle fibers are formed by the tubulin protein. They are the spindle fibers found by the tubulin protein. Okay, so you will have the spindle fibers at the end of the division. Sorry, at the end of the prophase, and these spindle fibers are attached to the chromosome. Okay, they will attach to the chromosome. I'll see how they look like. Sorry. Okay, and we have centriole here, we have centriole here, and these centrioles are forming the spindle fibers containing the chromosomes. Okay, so they will contain chromosome like this. They will contain chromosome like this. Chromosome, chromosome, and we have here is your centromere and kinetochore. Remember these two things: centromere and kinetochore. Okay, so you can see in this picture there is the irregular arrangement of chromosomes. Irregular arrangements of chromosome. Irregular arrangement of chromosome. There is the irregular arrangement of chromosome. So all the changes are here in the first phase of the M phase, that is the prophase. Please see again and try to remember this thing. Next phase is so simple, that is the metaphase. Okay. Next is the metaphase. Second stage is the metaphase. What is the metaphase? The end. The metaphase marked the start of second phase of mitosis. This is the second phase of mitosis. Second phase of mitosis. In this phase, complete dissolution of nuclear envelope. Okay, chromosomes are chromosomes are clearly visible. Chromosomes clearly visible. That's why the metaphase is the stage. If you want to study the morphology of chromosome, can be easily studied. Morphology can be easily studied. We can. This is the most important point about this. Okay, they will ask you in the NEET exam. Second thing, the metaphasic chromosome made up of the made up of the two sister chromatid. How? Okay, so this is the chromosome and they are the two sister chromatid, one and two. There is sister chromatid, sister chromatid. Okay, now I told you earlier that at the center of the this is the centromere, okay, and the centromere has the kinetochore. So this is the place where spindle fibers comes and attach, provides attachment to the spindle fiber, provides attachment to the spindle fiber. Okay, now next thing, should the metaphasic plate, so, okay, sorry, at the metaphasic stage, at the at the end of metaphase, what we will observe under the microscope. So you will observe all the chromosomes align at the aligns at the equator and known as the also called as metaphasic plate. Metaphasic plate. And second thing, spindle fibers attached to the kinetochore of the chromosome. Spindle fiber attached to the kinetochore of the chromosome. So it will be this stage you will get a cell like this. Here you will have the centromere, sorry, not centromere, centriole, and you will have some chromosomes here. Okay, and this chromosome will have the kinetochore over here, and we have spindle fibers from the centriole will attach to the kinetochore, attach to the kinetochore, attach to the kinetochore, and again centriole, kinetochore. You can see all the chromosomes are aligned at the equator. They're aligned at the equator. The equator. So this is known as the metaphasic plate. Metaphasic plate. And these spindle fibers are attached to the kinetochore. Please see here this thing. They are attached to the kinetochore. Okay, so they are attached to the kinetochore. Okay, so this one is the attachment. Okay, so here is your centromere, and this is your kinetochore. Kinetochore and spindle fiber attached kinetochore. Okay, so this is stage completed here. We will get a structure like this. Okay, I am drawing its structure. Okay, so please, and this is the structure of uh, chromosomes, full structure of prophase first, and again this is the structure of metaphase. Okay. Next, third stage is the anaphase. Third stage is the anaphase. So anaphase, at the anaphase, at the onset of the anaphase, each chromosome arranged at the metaphasic plate. So basically, this is the initiation from this cell. There will be initiation of the anaphase. Now, the chromosome present at the equator pole. The chromosome present at the equator pole like this. And here is your spindle fiber. Okay. Now there is shortening of the spindle fiber. Shortening of spindle fiber. So what will be the consequence of the shortening? The consequences of the shortening is the increased tension over this point. Tension over this point will increase. Okay. So due to this, the chromosome split simultaneously after the tension increases at the from the centromere. Okay. And they will have two sister chromatid. Okay, they will have each spindle fiber will have two sister chromatid. Okay, how it will look like, I will show you after some time. Okay, so at the end of the anaphase, at the end of anaphase, at the end of anaphase, centromere split and chromatid separate. First thing, centromere split and chromatids separate. Second thing, chromatids move to opposite pole. Chromatids move to opposite pole. Let's see in the diagram. So they will contain the sister chromatid like this, and again there will be shortening of spindle fiber. There will be shortening of spindle fiber. So centromere splits and chromatid separate. Now centromere split and chromatid separate. Chromatid moves towards the opposite pole. They're moving towards the opposite pole. So this will be the structure at the anaphase. Now there will be last phase of the M phase, that is telophase. Telophase. Now the telophase. The telophase chromosomes cluster at the opposite pole. Spindle and their identity lost as a discrete element. Okay, so first thing will be there. Chromosome cluster at opposite pole and identity lost as discrete unit or element. Discrete element. Second thing, nuclear envelope assembles. Nuclear envelope assembles around chromosome. Around chromosome. Our aim was to separate the chromosome, to duplicate the chromosome, and aim is completed. Now we have to come into the initial position, like the cell has the nucleus and all the things. Okay. And nuclear envelope assembled around chromosome. Nucleolus, Golgi apparatus, ER, endoplasmic reticulum, reform. Okay, so it will look like this. How? See again here. Okay, so we have cell here. Okay, and we have centriole, and here the chromatist comes and lost their identity. And again, there will be the reformation of nuclear envelope. Reformation of nuclear envelope. The nuclear envelope, double membrane nuclear envelope, and ERs, Golgi apparatus, and nucleus also forms. This is the telophase. So basically, we divided the nucleus. Here the nuclear division is completed. Now, we told you there is the two things. One is the nuclear division, and again there will be a cytokinesis. So we will reach to the we reach to the this phase, cytokinesis. We completed the cytokinesis of the mitosis. Now we will carry out the cytokinesis. Now cytokinesis. Cytokinesis. Now cytokinesis of two type. I told you, cell furrow method and cell plate method. So cytokinesis in animal cell. Okay, so in the animal cell, we have again nuclear centriole and this nuclear membrane, here your chromatin material. Okay, and now There will be in this animal cell there is the furrow method. Furrow method. In the furrow method, there will be development of the appearance of the furrow in the plasma membrane. Furrow look like this. Okay, furrow look like this. I am showing you a furrow. It will look like this. It will look like this. This furrow will move toward the center. It will move toward the center, toward the center, toward the center. At the center, they will meet each other. They will meet each other. So, and they will, the furrow will meet each other at the center, and ultimately they will divide into two different cells. At the nucleus, chromatin, and we got two cells. Okay. So the movement of cell furrow is the towards the center. Okay, so towards the center, this is the centripetal movement of furrow divides into two daughter cells. Then into the two daughter cells. But in plant cell, there is a different thing. Why? Because cell wall is the hard. Cell wall has the hard rigidity, and it is inextensible. Okay, so in this plant cell, we have another method, that is the cell plate formation. Cell plate formation. Okay, so the plant cell, sorry, plant cell, and we have two cells here. Now, cell plate will form at the center. Okay, cell plate will form at the middle, and they will move centrifugally like this, and it will cut off the cell into two total cells. The cell plate move centrifugally, centrifugally, and basically cell plate is a look like middle lamella. Cell plate look like middle. What is the middle lamella? It is the common wall between the two cells. Common wall between the two cells is the middle lamella. It will look like the cell plate. So cell plate moves centrifugally towards the periphery, and it will divide the parent cell into the two daughter cells. So that's how the cell division takes place. Now, most important thing in the mitosis. What is the significance of the mitosis? In the mitosis, the chromosome number remains same. How it is possible? For this, please see here. We have three chromosomes here. How will you count the number of the chromosome? Number of chromosome is equal to number of centromere. Okay. So 1, 2, 3 centromere, 3 chromosome. See again here, 1, 2, 3 chromosome. For 3 centromere, 1, 2, 3 centromere. This is the 3 chromosome in this cell, 3 chromosome in this cell. So the number of the chromosome in parent cell, number of the chromosome in the daughter cell is same due to the N of it. Okay. So it is about the mitosis. Basically, mitosis takes place in the all. It takes place all somatic cell, body cell. Okay. increase number of the cell, increase number of cell, takes part in the healing of wound, any wound healed by the mitotic division, because it is uh, only the cell division which increase the number of the cell. Healing of injury, healing of injury. Somatic cells means that from all somatic take place in all somatic cells. Somatic cell means body cell, 2N cell, diploid cell. Okay, diploid remains diploid. Diploid body cells, all the body cell is diploid. Sex cell is haploid. Okay, so it is about the mitosis. So we completed the mitosis, and uh, one more cell division remaining is the meiosis, and we will see it in the next lecture. It is enough for today. So please see once again about today's lecture. So we completed the cells, fundamental unit of the life, chromosome, types of the chromosome again, and telocentric, cytoskeleton, cilia, flagella, and central centrosome. Then I told you about the microbodies. In short, cell cycle and cell division, cell cycle. This diagram is most important for the NEET. Please refer your NCRT for this diagram. You will see different type of the questions them they are asked from the cell cycle. This diagram particularly. Okay, so please see the diagram, and again division of the uh, classification of the division. G1 as G2. This thing is most important, and G2 phase. Then I told you about the differentiation, de-differentiation, redifferentiation. G note phase, the quiescent phase. Then I told you about the M phase, four phases: prophase, metaphase, anaphase, telophase. Prophase is about the condensation of the chromosome. Then there are the different diagrams you can see, and you can understand the processes taking place inside the cell division of the mitosis. Okay, so thank you and have a nice day. Any doubt you can ask now. Any doubt?
No, sir.
ओके थैंक यू एंड हैव अ नाइस डे
Thank
समझ
you.
समझ में आ रहा है आर्यन
हाँ सर.
ठीक चलो हैव अ नाइस डे और पढ़ते रहो क्वेश्चन सॉल्व करते रहो इसी चैप्टर के प्लस सेल के फ्रॉम द स्पीड लैब एंड कोई डाउट